Welcome back to our video on meiosis. Today we're looking at the steps that we take in making those sperm and eggs. They are similar to the steps of mitosis, but slightly different. So we'll be going over the differences between each. First off, just like the other cell division, meiosis does not refer to absolutely everything within this. Meiosis is not all of cell division. We still have interphase and cytokinesis. So the first thing that happens, as always, is interphase. And in interphase, as we learned previously, you get G1, S, and G2, where we grow, copy DNA, and prepare for further cell division. What that looks like is, as previously, we have our little cell, and we have copied our DNA. Now, to make this a little bit simpler, we have 4 different chromosomes this time. As always, we have 2 of each color. So we have 2 red, 2 orange, 2 blue, and 2 green. That one is smaller. If you have trouble seeing that one, for meiosis, we're going to make the cell bigger, so you will get to see how we have each of them. Next, in meiosis, as I said, the phases are similar, but not exactly the same. Meiosis starts with prophase 1. Same as mitosis, you get your centrioles, you get your thicky boys, your chromosomes are condensing, and you also get the nucleus disappearing. Same as mitosis prophase. However, you also get 2 other things. Your chromosomes pair up, homologous, as we said before. And with those homologous pairs, you also get a thing called crossing over. So it's the same as prophase in mitosis, but also has a couple of extra steps. So let's draw what that cell would look like. We have this cell. Our nucleus is starting to disappear. We'll make that nucleus a little bit larger so that you can see exactly what's inside of it. It wouldn't normally take up this amount of space, but it'll be easier to see. And then we become our thicky boys. So let's make our thicky boy orange, our thicky boy blue, thicky boy red, and our thicky boy green. 4 different colors. And we will also get our centrioles. Now for this, since we're already using orange, our centrioles are going to be a sort of purpley magenta color. But I also said that they pair up. So for this, we're going to pretend that blue and orange are homologous, so they will pair up. And that red and green are homologous, so they will pair up. And when they pair up, we do this crossing over thing. Crossing over means you take those like genes, that like information, and you swap it between them. Since they're homologous and you find these same parts in that same area, they can swap information. They can do this hundreds of times. But to make it easier on us, we are going to make it not happen 100 times, but we're going to make it happen a little bit, only once. So this green and this red are homologous, and when they cross over, that portion of the green and that portion of the red swap locations. And the same thing will happen between the blue and the orange. They have swapped information. That is this process of crossing over. Once we have finished that, we can move on to metaphase 1. And in metaphase 1, we line up like in prophase, but we are in our homologous pairs still. We are still in our homologous pairs. So when we line up, we have not broken from what we see over here. So we line up in the middle, same thing as the previous mitosis. But when we line up, we are lining up still in our homologous pairs. We are side by side. And then same thing as mitosis. You'll get centrioles lining up on either side, and get your spindle fiber attaching. However, you'll notice they only attach to one side of each chromosome. That is important because when we move to anaphase 1, we only separate the homologous pairs. We are not breaking those X's apart. So when we pull them in different directions, we don't separate the X's that have formed here. And I am simplifying them into just the simple X's to save time on this. So this is what you would see in anaphase. And finally, you will have squeezed over here telophase 1. We can kind of erase this so that you don't get confused for that. We'll even just move the line over. Telophase 1. we undo prophase 1 pretty much. So chromosomes condense, nucleus disappears, centrioles appear. We're undoing that. And we're going to separate, and we are reforming that nucleus, and we are uncondensing. But one thing you'll notice about our uncondensed, when you look at it, you will see that we still have crossed over. There's still a little bit of orange in this blue, still a little bit of green in this red. That doesn't change when we uncondense. You will still have those differences. Same thing here on the left cell, or the left cell that is going to be produced. You have a little bit of red in that green, and a little bit of blue in that orange. And then you would have cytokinesis, try to balance these out as even as possible. We are just going to skip that since we covered it in mitosis, because it would be the same of just try to balance everything out. We move on over here, and we're going to move on to prophase 2. We do have a small little interface, however, the most important thing is moving into meiosis 2. Is you do not copy the DNA. DNA is not copied before meiosis 2. And then you get prophase 2, metaphase 2, anaphase 2, and telophase 2. And they do generally the same that they do in mitosis. So you'll get your centrioles, you're condensing your chromosomes, and your nucleus is disappearing. And since we formed 2 cells over here, as you can see in telophase, we would have formed our 2 cells in cytokinesis. In prophase 2, we are doing this amongst 2 cells. But we did not copy our DNA. So when we condense, we're not going to condense into 4 chromosomes in each cell. Instead, we are only condensing into what we have in each cell, which is green with a little bit of red, orange with a little bit of blue, because that's what we pulled into this cell. And same on the other side. It'll be blue with a little bit of orange, and red with a little bit of green. Then when we move to metaphase, it'll be, you line up in the middle, but orange and blue are not in the same cell. Green and red, not in the same cell. So we are not in homologous pairs. We are lining up in the middle, just like in mitosis. just like in mitosis, they are not side by side. They are in a line, up and down, since they are not homologous pairs. Green, if this is chromosome pair 1 and this is chromosome 2, green and orange, 1 and 2, don't have the same information, so they can't be side by side. They're in a line, like in mitosis, where you don't have copies. Then we move to anaphase 2. We pull them apart. But we are pulling the chromatids apart, since each of these would be sister chromatids, which we learned in our very first video, in our cell division definitions. If you need to go back and look at that video, feel free to. But these are the sister chromatids that we are pulling apart here. And I didn't draw the centrioles in here. I should probably do that because those are reforming. We'll just quickly do that in magenta because this video is becoming very long. So we have those there. We have our centrioles on each side of the cell. And same thing in mitosis and in meiosis 1. You would have those spindle fibers leading from the chromatids to there. Then we pull those apart. Whoops, I have drawn that wrong. I mean, it doesn't really matter, but keep left cell, right cell, right cell. We'll go ahead and keep it exactly the same. And then this one will be a little bit there and a little bit there with a tiny bit there. So we have separated those sister chromatids by pulling them apart. And finally, in telophase, we start to form two different cells. Same thing that you see up here, except we are undoing prophase 2. We are undoing what happened here. And we are forming those two cells. So let's see if I can do this quickly. We'll put this cell up here. So we have a little bit of green up here. And then we have over here a lot more green and a large amount of green with a little bit of red. Large amount of red here, and a decent amount of red there. We then have our blue with a little bit of orange, our full blue here, and our little bit of blue there, filled with full orange, a large amount of orange, and a little tip of orange. So you can see how when we end, this would end with cytokinesis, again, dividing them. We're just going to put end. We end with 4 cells. Mitosis, you saw, ended with 2 cells. This one ends with 4 cells. Let me draw the nuclei back. And we have blue and red. little bit of each. Here, with tips there. Got green and orange. So if you look at each of these cells, full blue, full red, mostly blue, mostly green, mostly orange, all green, all orange. All of them are genetically unique. That is why meiosis is important, because these are haploid, which means, unlike what we started with, where we have 2 copies of each chromosome, we only have 1 here. So when you combine this, if it's a sperm, if you combine it with an egg, if it's an egg, you combine it with a sperm, you will get 2 copies of those chromosomes and make a diploid. It's what allows your parents to have 46 chromosomes, and you to have 46 chromosomes, and your kid to have 46, and your grandkids to have 46, because you always divide before you multiply. You divide into half, multiply by 2, so you always stay at 46. Go to 23 for your sex cells, kid has 46. That kid goes to 23 for sex cells, goes to 46 for their kid. And it repeats on and on and on. So hopefully this series has made it easier to understand what is happening in cell division, in mitosis and meiosis. That is it for this cell series. The next series that we will be looking at is what exactly these are. Not just chromosomes, but what is DNA? We said that it was the instructions for the cell. What instructions does it contain? How does it store that? And how does it write messages to instruct the cell, which is called RNA. So that will be our next video series.